Hello, everyone. Thank you for joining in. This is a real pleasure to introduce Akem's Gough talk today. Akiem grew up in Jamaica and then did his bachelor degree in biology at Florida Southern College in Lakeland. He got to learn about Archbold Biological Station through a field ecology lab that he participated in 2020, where he visited the main station, which gave him the idea to apply for an internship at Archbold. He's been working for Archbold for about close to a year at this point. He joined us in June 2021, and initially he was hired to participate in a West Indian marsh grass study that we have currently going on the ranch, but he ended up working on so many other projects here that we have currently. He has always been very good at accepting any task we gave him, whether it's like a very boring task like grinding forage or like acid washing in a chem lab. He always was very happy to do so. And he's always been a very pleasant person to work with in the field. We had some interns that bring like a radio with them. Akiem didn't need to do that because he would sing most of the time, so which is very nice when you do field work outside. So without further ado, I'll let Akiem take the stage.
Thank you for that wonderful introduction, Greg. And thank you for everyone for tuning in. And let's get right into it. So my project was on the seed bank persistence, seed output, and long-term seed viability of Hymenachne amplexicaulis. Hymenachne amplexicaulis is the species name for West Indian marsh grass. It is a Category 1 invasive species, and this means those are those plants that displace or hybridize with native species or alter ecological functions or structures, therefore changing plant communities. Specifically, West Indian marsh grass displaces Panicum hematomen and Echinochlora walteri, which along with the functions of water remediation and soil stabilization, they provide habitat for various fauna, including resources for feeding, breeding, and shelter. West Indian marsh grass also blocks waterways like canals and ditches and forms dense mats above and below water that may reduce light and oxygen, thus impacting aquatic populations. West Indian marsh grass has effects that are both concerned with both species diversity and habitat diversity, and thus has the potential to halt wetland restoration efforts. Now, what are the current control methods? There are many and varied, but the main one is herbicide through glyphosate and imazapyr. And they have their different advantages and trade-offs, but in general, when dealing with wetlands and herbicides, it's important to consider the effects of soil and water contamination. So there are also some proposed biological controls, such as Ichnodemus variegatus, this insect that feeds on West Indian marsh grass. Studies are still being done on its host specificity, though, to make sure that it doesn't eat other plants. And interestingly, cattle also present a proposed biological control because West Indian marsh grass is highly palatable to them and has high forage quality. Speaking of which, there is an ongoing study, the NRCS Exotic Control Project at Archbold, the Agroecology lab, where they're looking to compare the effects between grazing, herbicide, and the combined effect on West Indian marsh grass, and it will come up later in the presentation. Now, there are some gaps in knowledge about West Indian marsh grass where if we fill them, we would be able to help form even more efficient control strategies. First of all, there's seed bank persistence. Concerning the seed bank, this is a portion of the soil that has viable seeds after the first germination season. And I'll show this in a visual right after this. A transient seed bank has seeds that are viable only until the next germination season, and this is usually less than a year after the seeds are produced. And then a persistent seed bank has seeds viable until at least the second germination season, so beyond a year. And some seeds are even persistent above 5 years. There's also long-term seed viability that we'd like to look at, and it's not just the seeds in the soil, but the seeds themselves how long are they viable for? And a viable seed can be considered to have living and respiring cells in their embryo. Now, this is the visual I mentioned. So Western marsh grass produces and releases seeds around August to September. And once the water level decreases in the dry season, the seeds get access to soil, and that's when the first germination season occurs, the first opportunity for seeds to germinate. Now, there might be some seeds left back in the soil, and they would get the chance to incorporate into the seed bank. And the question would be, is that seed bank persistent? And to answer that, one would look at the seeds from approximately a year after and see if those seeds are able to germinate. Now, there is a study in Australia. You can see the areas on the map where they managed for West Indian marsh grass that actually did study seed viability of West Indian marsh grass, and they found that it was 96% after 2 months and 78% after 4 months. They also did a seed burial trial where they planted seeds at different depths and then took them out at different periods of time to test viability. And they found that the seed bank is persistent, with 21% of seeds on the surface viable after 6 years, as you can see on the graph, and then 8 to 24% after 8 years. And you might be hearing this and wondering why I'm saying there's gaps in knowledge. Well, it might be that these results are specific to Australia. They did not specify just from the paper. And interestingly, we have done studies at Buck Island Ranch, and their experiments with germination trays in areas that you would find West Indian marsh grass. However, we found very low West Indian marsh grass germination. I wasn't here yet, but I say we as the Agroecology lab. And this contradicts with the study in Australia, of course, possibly due to differences in location or other factors we're not aware of. Nevertheless, the results from our experiments are unpublished and wasn't specifically aimed at West Indian marsh grass. So I think doing so would make it more comparable to the Australian study. Which leads us to my aims, which are to fill those gaps in knowledge about West Indian marsh grass, seed bank persistence, and long-term seed viability here in Florida. And also to estimate seed output to get an idea of the scope of the effects of these factors. And I thought it would also be interesting to compare the seed output between the grazed and fenced wetlands because of what I mentioned about the cattle and the West Indian marsh grass. So in doing so, we hope to help inform efficient control strategies and explore if cattle grazing could be one of those strategies. Now, looking back at the timeline in context of my aims, after seeds are released and the germination season, I would ideally collect soil approximately a year after, asking the question, will those seeds germinate? And then once seeds start producing, I collect them, ask how many are produced and grazed and fenced, and how many are viable, and for how long do they stay viable for? Leads us to our hypotheses. First of all, if West Indian marsh grass forms only a transient seed bank, then only a few seedlings will germinate from soil collected a year after the first germination season. While if it's persistent, then many seeds will germinate within that same timeline. And I predict that it will be transient based on the previous experiments. Regarding long-term seed viability, if time has no impact on West Indian marsh grass viability, then the viability will not change as time progresses. While if it does have a negative effect, viability will decrease significantly as time progresses. And I predict specifically that viability will decrease over a period of 4 months, which should be comparable to the timeline of the results of the aforementioned study. Now, regarding comparing between grazed and fenced wetlands, if grazing has no effect on West Indian marsh grass seed output, then seed output will not differ between grazed and fenced wetlands. While if it does have a negative effect, seed output will be lower in grazed than in fenced wetlands. And I predict that it will be lower in the grazed wetlands. Jumping into our methods, our study site is Buck Island Ranch. It has numerous seasonal wetlands. I compiled data from numerous years to see the ones that had the highest West Indian marsh grass incidence. And I chose 7 of them, as you can see circled on the graph on the right, as well as on the map I created on the left. And these are the areas that I did all my data collection. Starting with the seed bank sampling. I use a 1 meter square quadrat, which looks like this, and I tried to go to areas that appear to only have West Indian marsh grass cover. And then I used a soil core that looks like this. Here are the dimensions. I went to 20 centimeters just so I can get enough soil. And then there were two plots in each wetland, so I had subsamples. After air drying the soil samples and homogenizing them, they look like this image on the left. And I removed roots to make sure that any germination was from seeds. And then I also sift the soil and remove larger materials so it's easy to spread in the trays. After preparing the trays, they were held in a shade house where they had an automatic irrigation system. And I checked germination 2 to 3 days per week. And I stopped when there was no germination for 3 weeks. And then throughout the process, I recorded and removed seedlings as soon as I was able to identify them. Now for the seed output. The main idea is to get the number of inflorescences in a 1 meter square plot like this, and then the number of seeds from an inflorescence, which is a flower head or a seed head, by the way. Right now it's in the stage with the florets and they'll all develop into seeds. And then I'll get the number of seeds in this whole plot. So to get that, I went to the same 7 wetlands, had 2 plots, and I counted all the inflorescences. Then I collected some, and instead of counting all the seeds, I had to count some and then measure the mass, and using a formula, I estimated the number of seeds. I didn't count because when I started, I realized there was a lot of seeds that come from just one inflorescence. And this is how I came up with that formula, and I can go back to it if anyone wants to see. And then I had to divide by 2 because 2 inflorescences were collected per bag, and I wanted seeds in 1 inflorescence. And then we get back to the main formula, which tells us the number of seeds produced in 1 meter square plot. Now with the comparison, I knew the wetlands that were fenced and grazed, so I separated them and then compared them by seed output per plot as well as seed output per inflorescence. Then there's also the NRCS Exotic Control Project I mentioned before. I combined data with that project to compare inflorescence count as well as inflorescence length. And I was interested in inflorescence length, especially because when I was collecting data, I noticed that the inflorescences were shorter in the grays than in the fenced wetlands. So I wanted to put that in data and statistics. And I used linear regression modeling in RStudio for statistics. Now for the long-term seed viability. I collected seeds from 3 wetlands this time and 20 from 20 inflorescences from each wetland. And okay, this might be a lot of words, but if you look at the image on the bottom right, you see stacks of Petri dishes. Each stack represents a treatment group, and each Petri dish would be a replicate, and within each Petri dish is 50 seeds. And the treatment groups represent holding times, that is, amount of days the seeds were held in the fridge after they were collected. And as you see, it goes from 1 to 120, separated by 15 days. So there were 9 treatment groups. Each time that I tested the seed for viability, I had to precondition it first by soaking it for a certain amount of time and piercing every individual seed. And then I applied the test. It's a chemical dye called tetrazolium for short. And once the seeds get time to soak in them, if they're viable, then the embryo will appear red. And this image shows the proportion of the embryo that has to be red before it can be considered viable. So everything on the right with the negative is not viable. And I also use linear regression modeling in R for the statistics. And here's an in real life example of how the seeds look when they're empty, viable, and non-viable. Now let's get into the results and discussion. First off, starting with the seed bank sampling. On the left of the graph, you see the percentage of West Indian marsh grass seedlings, and that corresponds with the green bars. As you can see, it never surpassed 10%. And then on the right, you have number of seedlings. Orange is total seedlings, and yellow is only West Indian marsh grass. Sometimes you're not even able to see the yellow because it's that much lower, and it was never noticeably, it was always noticeably lower than the total number of seedlings. So we deduced that the seed bank was transient, with only 2.27% accounted for by West Indian marsh grass, which equals 51 out of almost 3,000 seedlings. So this is similar to the findings of the previous experiments, but why might this be? It could be that the West Indian marsh grass has low seed output, which would mean there are not many seeds in the soil to begin with to germinate. Or there are many seeds, but they're not viable, or the viability decreased over time. It is important to consider, however, that a viable seed does not mean it will necessarily germinate. And there are certain conditions and abiotic factors that might limit the seeds from germinating, especially past the first germination season. West Indian marsh grass also grows from stones, which look like this, and they can also be considered a dependable reproductive strategy. So it is entirely possible that instead of investing energy into seeds to be viable and germinable for a long time, West Indian marsh grass invests energy into stolons. Now, to test if seed output and seed viability were factors in why we found a transient seed bank, we can look at our other results. For seed output, it was found to be generally high. Ooh. Sorry. Oh. So as you can see on the bottom, the number of inflorescence is high, number of seeds, as well as number of seeds in plot and number of seeds per inflorescence as well. And this makes sense because previous studies have described West Indian marsh grass as a prolific seed producer. And even the study from Australia found more seeds per inflorescence than us, less inflorescence per meter square, and then up to 100,000 seeds when you calculate it per plot. And this is less than ours, and theirs was considered high, so we would consider our value high as well. Now, considering that wetlands have abundant nutrients and other essential materials, and that West Indian marsh grass is invasive, it isn't a surprise that it would capitalize on the access to those materials in order to produce many seeds. So we can rule out low seed output as a factor why we found a transient seed bank. Now let's look at long-term seed viability. As you can see on the graph, it's decreasing, and this decrease was found to be a strong evidence of decrease. However, we deduced that it was at a low rate. As you can see, there's a point where it went back up, and even at 120 days, it was still above 50%. Comparing this to the study mentioned before, there was 78% after 4 months, and we aren't that different. We can also compare to the seed burial trial, the lab version of that in the Australian study, and theirs was even higher. Looking at a longer time frame, theirs even went up much higher. So it's not far-fetched that our seed bank decreased at a low rate. It is interesting, however, that ours started decreasing before theirs. Nevertheless, we can rule out low seed viability as why we found a transient seed bank. So looking back, we've ruled out low seed output and low seed viability, as well as viability over time. So it is possible that the conditions are not favorable for West Indian marsh grass to germinate from seeds past the first germination season, and or that West Indian marsh grass is investing energy into stolons instead. Moreover, since the seed output is so high and viability is so high, especially to begin with, maybe the seedlings produced from that first germination season are sufficient and have spread successfully. Now let's look at our comparison between grazed and fenced wetlands, starting with seed output per plot, which is the number of seeds, not just in one inflorescence, but in all the inflorescence, or an estimate of, and comparing it to another plot. We found fairly strong evidence that grazing reduced the seed output per plot, after log transformation. And you can see on the graph that it's lower in grazed than in fenced. Similarly, we can look at inflorescence count. And we found strong evidence as well that grazing reduced inflorescence count per plot after log transformation. And you can also see that it's lower in grazed. And then for seed output per inflorescence, which is the number of seed in one inflorescence, we found fairly strong evidence that grazing reduced seed output even before log transformation. And it's even clearer on the graph that grazed is lower than fenced. And then this one is the most clear. This is inflorescence length, which is self-explanatory. And we found very strong evidence that grazing reduced inflorescence length, as you can see very clearly on the graph where grazed is lower. Now, we found that all those metrics related to seed output were lower in grazed than in fenced wetlands, so cattle could be a potential control strategy. And also considering that when we saw the inflorescence lengths, they were not short because they looked bitten or chewed, they grew shorter. The effect might not just be mechanical, but also from physiological stress, which is interesting. And more studies would need to be done on whether cows transport the seeds, because that could negate the effect of grazing, or it could inform us to simply make sure that grazing occurs before seeds are produced. So to conclude, we found West Indian marsh grass has a transient seed bank, likely due to conditions past the first germination season that do not promote seed germination. And seed output and seed viability were found to be high and high over time and could have contributed to successful spread in the first germination season. And West Indian marsh grass also have stolons to depend on as a reproductive strategy. Additionally, cattle grazing was found to have a strong effect on West Indian marsh grass seed output and could be an efficient control strategy. Future studies would be good to be done on the seed bank, looking not just a year after, but right after the first germination season and a month after, 2 months after, and so on, to see at what point the seeds start and stop to become germinable or able to germinate. And it would also be interesting to determine what kind of factors limit the seeds from germinating after that point. I also think it would be good to do a growth chamber experiment that's similar to the viability, except the seeds are in soil, to not just see if the seeds are chemically viable, but able to germinate after those same periods of time, and maybe even extending it over a longer period of time, so it's even more comparable to the Australian study aforementioned. And also to compare the rate of growth between stolons and seeds to see which reproductive strategy is most efficient. And also, as I mentioned, to investigate if cattle transport seeds. Wow, this did not look like this before. But I would like to thank Dr. Betsy Boughton and Dr. Gregory Saunier for assisting me throughout the project and giving me guidance, as well as the entire agroecology lab for being so supportive and helpful in finding materials and ways to communicate this study. And I would also like to thank the USDA NRCS grant for funding my project and my stay here. These are my references, and we can be open for questions now. Thank you.
Thank you, Akiem. So we do have a few questions, but first I actually forgot to mention something important in my presentation of Akiem earlier, and that thing is that Akiem is going to remain to work with us as a research assistant in the future, so we are pretty happy about that. So let's go back to the question. So I guess Lydia, you answered Lydia's question during your presentation, so it's good. So Betsy has a question. Great job, Akiem. Have you observed West Indian marsh grass seedlings in the field? Now that the wetlands are dry, I am wondering if you saw any as they dried down after seeds were dropped.
I definitely have seen Western Union marsh grass seedlings, especially when I went to go check wetlands the other day. But it's hard to tell if they're growing from the stolons or from the seeds. And I didn't bother to dig to the soil to disturb it, but it's entirely possible.
All right. We do have another question from a previous Buckeye Lone Ranch intern, Flynn Hibbs. Fantastic presentation, Akiem. Has there been any studies on if grazing or disturbance influences numbers of stolen in West Indian marsh grass?
Not that I know of, but with the NRCS project with which Greg and I do, Greg has collected stolons, and we are looking at the effect that position of the stolons have, as well as um, if the cattle grazing affect the stolons as well. So that could be something we could answer in the future.
We have another question from Buckeye Lone Ranch intern, Maya. What flavor juice will you drink to celebrate your incredible presentation?
I'm thinking of like a white berry mixed with lemonade, and a little Sprite. Thank you.
I guess that's all the questions that I have here. And a lot of people commented that your presentation was great, Akiem, so good job.
Thank you. Thanks.
All right, so that's it.
Yeah, okay. Thanks so much, Akiem. Congratulations on your job.
Thank you so much.
All right. Thanks everybody else for attending today. Please watch our website and newsletter and Facebook page for information because we have a lot going on in May that I haven't been able to compile yet, but we have so far 3 presentations coming up in May. So stay tuned. We'll see you soon.